Hello everyone. I hope you can see my screen. My name is Tagenya Shparo. I'm your lab professor. Today I'm trying to record uh, the slides on cell structure and cytoskeleton. And in your textbook, I think it appears as chapter 4. I'll try to go on them like really quickly because everything I'm going to record is already on the slide. So this is just trying to give you a highlight of what might be on your quiz. And probably this could be a repetition from your lecture slides. Right, cells under the microscope. Cells are extremely diverse, starting from bacteria all the way to eukaryotic organisms like us. They are extremely diverse, and nearly all of them require a microscope. It is really difficult to see any regular cell with a naked eye. The only cell that we could be able to see with our naked eye is probably the human egg. Each type of cell is in our body is specialized for a particular function. It's difficult to imagine two cells performing a similar function. There are cells that perform a similar function, but usually under normal circumstances, each cell organized as a tissue performs a certain kind of function. We observe cells under two kinds of cells, usually under the light microscope and electron microscope. For the light microscope, as the name implies, it requires light to observe the cells. For the electron microscope, we just need a beam of electrons reflected on the cells to look at how the cell is organized into tissues and everything. These are the cells under microscope. Yeah, as you can see with the electron microscope, we can actually be able to see anything from the atoms all the way up to the human egg. And for the light microscope, with light microscope, we cannot be able to see viruses. Viruses can only be seen through electron microscope. Why are cells so small? The reason why cells are so small is because small cells actually have more surface area for exchange compared to large cells. Therefore, through evolution, cells become so small. With cells need surface areas large enough for entry and exit of materials, and that happens only when the cells are small. And we need a larger surface area to volume ratio. Small cells have more surface area for exchange, we said that already. And through adaptation and evolution, the surface area of most known cells is increasing. One simple example is the microvilli in small intestine, which increases the surface area of absorption of nutrients. As you can see here, this is 1 4 centimeter cube, and the whole surface area is the surface area of these 6 sides, the one on top, at the bottom, front, the back, and the left and right. If you can calculate the area, that's the whole surface area. The volume is just the multiplication of the lengths, the widths, and the height. The lengths, the height, and the widths. So for this one, the surface area is smaller compared to, for example, say this one. One simple way to see the difference is, let's say somebody asked you to paint all these. To paint this 1 4 centimeter cube, you just paint this side, the 6 sides, and that's it. But if somebody asks you to paint all these, it might actually take 64 times more paint than this one, because you paint this side and then you paint this side and then you paint on the sides. It's like this one and this one are already part of this one, but The rest, the left and the right, the back and the front, this is new. It's like you can't try it at home. You can't just take any large cube and then try to make a smaller cubes out of it and then try to paint it. When you think of the cell structure, the first part that appears is the plasma membrane, which marks the boundary between the outside and the inside of a cell. This is like any regular boundary for any nation. And there's a regulation as to what can get in and what can get out. And this layer is made of phospholipids, and it's called phospholipid bilayer, and it has a lot of proteins embedded on it. The phospholipid bilayer is made up of polar heads and non-polar tails. The heads are hydrophilic and they are usually pointed outwards, whereas the non-polar tails, the hydrophobic ones that are afraid of water, are pointed inwards. This whole structure was proposed as in the fluid mosaic model, because there's a mosaic but it's a fluid. And this is how it looks like. This is the polar head, which is hydrophilic, and this is the non-polar head, the yellow part. And the polar heads at the bottom are also pointing inwards. And everything you see, the blue ones are proteins. This is, for example, a protein molecule serves as a protein channel, or yeah, this is just a protein. There are various membrane proteins or proteins that are found on the cell. One of them is the channel proteins. Channel proteins, they don't change their shape. They are just a tunnel, and only certain kind of molecules that can be able to fit through that channel can get into and outside of the cell. The other kind of proteins are transport proteins. For this, they need energy. Unlike channel proteins that do not need energy, transport proteins actually need energy. Yes, and they don't usually change their shape. Cell recognition proteins, as the name implies, this one, cell recognition protein, its task is to identify or to distinguish between our own cells and the cells of other organisms. And this has a protective function, protects us from invaders. Receptor proteins are proteins that are really relevant in downstream pathways, because, for example, if this is a signal molecule, it interacts with the receptor protein, and their interaction actually initiates a downstream pathway that you might learn in, let's say, biochemistry or molecular biology. Enzymatic proteins. Enzymes are made up of proteins, and as you know, enzymes reduce the activation energy of any kind of chemical, not any, just specific kind of chemical reaction, and they directly participate in metabolic reactions. And in fact, it is really difficult to think of any biological reaction without the help of enzymes. There are also junction proteins that form junctions between cells. This is really important for cell-to-cell adhesion and communication. Yes, because cells do not just exist by themselves. They are not independent. They have to interact with neighboring cells in a tissue or with even other cells and outside molecules. There are two main types of cells, and there is a cell theory that differentiates between these types of cells. And according to the cell theory, all organisms are composed of cells, and cells come only from pre-existing cells. You cannot get a cell without any pre-existing cell. We cannot get a fetus or cannot get a human being without the egg and sperm. All cells have a plasma membrane to regulate movement of material inside to and out of the cell. And the cytoplasm is where the chemical reactions occur. It's like It's like any kind of compartment. There's the compartment or the boundary, which is the plasma membrane, and everything else that happens within the cell is gonna happen within in the cytoplasm, because cytoplasm is whatever you found within the plasma membrane. In other organisms like bacteria, the chemical reactions might take place in plasma membrane instead of the cytoplasm. Also, all cells have the genetic material, which is the DNA, and it is relevant for our growth and production. And there are two main types of cells. And let me see if I could minimize this thing. There are two main types of cells. Based on the organization of genetic material, one is a prokaryotic cell and the other is eukaryotic cell. The biggest difference between prokaryotes and eukaryotes is that prokaryotes do not have membrane-bound organelles, or in particular, the biggest organelle is nucleus. So if you see their nucleus, they don't actually, some of them don't actually have a nucleus, rather they have a nucleoid region that contains all the genetic material they need, but that nuclear region is not bound by membrane. Even the rest of the organelles are not bound by membrane. For eukaryotes, everything has a membrane. Almost every single organelle has a membrane. Okay. Prokaryotic cells they include organisms from the domains bacteria and archaea. Actually, the two prokaryotic kingdoms domains are domain bacteria and domain archaea, and they are generally smaller and simpler compared to eukaryotes like us. The fact that they are small and simple enables them to reproduce very quickly and effectively. Because, for example, humans were bigger and were very complex. That's why we take 9 months to have a baby. But for the bacteria, they are small and simple, and they could have billions within that time frame. They are extremely successful group of organisms, and for example, bacteria, we know them. They cause some of them cause disease, and it is really difficult to imagine the world without prokaryotes, and their number is way more than the prokaryotes. They have a lot of crucial roles they play. Some of the bacteria actually we found in our guts, which helps in breaking down certain molecules we are unable to break. Some bacteria, they are found in food. For example, cheese fermentation happens through bacteria, and for plants are capable of capturing nitrogen from the atmosphere through the use of bacteria. Bacterial structure. Bacteria has a cytoplasm that's surrounded by plasma membrane in the cell wall, and the cell wall might have a capsule, which is a protective layer for the cell wall. But plasma membrane is the same for every organism we know. Every organism, living organism, has a cell, plasma membrane. Cell wall maintains the shape of a cell, and they do have a DNA, and it is single circular DNA. The reason why it is single circular is not the reason. It is single circular, but in cases of viruses, there are viruses that have a double circular DNA instead of single circular DNA. And the DNA is found in the nuclear region, not in the nucleus. As I told you, they don't have like an actual nucleus per se, but they do have a nuclear region, a region that looks like a nucleus, and the DNA, the single circular DNA, is found in that location. And bacteria do have ribosomes. Okay, if there is anything like I want you to memorize from this section, just know ribosomes. Ribosomes are the sites of protein synthesis in any cell. Ribosomes are the sites of protein synthesis. This is to mean that when the genetic material, the DNA, is transcribed or converted into a messenger RNA, that messenger RNA leaves the nucleus and it goes to the ribosomes where the messenger RNA is converted into an actual protein. That's why we say ribosomes are the sites of protein synthesis, or in another word, DNA translation or translation of messenger RNA. Bacteria has appendages, has the flagella, it has the fimbriae, and it has this thing is bothering me. It has the flagella, it has the fimbriae, and it has the conjugation pili. Flagella is for propulsion or for movement. Fimbriae is when the bacteria wants to hold on to something, or when they want to attach themselves to a certain surface, they use their fimbriae. And conjugation pili is their reproduction organ. They use it for DNA transfer. Yeah. This is how a prokaryotic cell looks, typically the bacteria. I want you to take a moment to look at the structures we have talked about. We have capsule, nucleate, ribosome, plasma membrane, cell wall, cytoplasm. Sometimes a picture is more descriptive. Eukaryotic cells, eukaryotes will have 4 groups of organisms: the protista, fungi, plants, and animals. They have a membrane-bound nucleus housing or containing the DNA. They are much, much larger than the prokaryotic cells. That's why we are able to see eukaryotic cells under light microscope, whereas the viruses were unable to see them through light microscope. They are compartmentalized and they contain organelles, like membrane-bound organelles, and we have 4 categories of organelles we need to see: the nucleus and ribosomes, the endomembrane system, energy-related system, and the cytoskeleton. We are going to see them. We are going to see every one of them. This is the structure of a typical animal cell. I want you to take a moment to take a look at these structures. This is the structure of a typical plant cell. Just take a moment to see what differentiates a plant cell from an animal cell, and what you can find in animal cell but you couldn't in plant cell. Just try to see the differences and similarities. Yeah, this is the structure of a typical plant cell. Now, the organelles that we find in eukaryotic cells. The first one is the nucleus and ribosomes. The nucleus stores genetic information, or in particular the DNA. The nucleus is organized into chromatin, and the chromatin contains diffuse DNA, like DNA that's not ready for transcription or replication. Then it contains proteins, some RNA, and then prior to cell division, DNA compacts into chromosomes. So just chromatin is everything that you find in the nucleus, including DNA, protein, histones. DNA is organized into genes, which specifies a polypeptide. Well, genes, for genes, first it starts from amino acids. And when you have 3 amino acids close together, they form a codon. And when codons come together, they form a gene, and those genes form chromosomes, and the chromosome form the DNA. And the DNA, upon its transcription, leads to the formation of messenger RNA. And this messenger RNA is translated into a protein or a polypeptide by ribosomes, or usually the ribosomes contain ribosomal RNA, and this ribosomal RNA is made in the nucleolus. This is probably the most visible part of the nucleus. The nucleus has a nuclear envelope, which is a double membrane system, and nuclear pores permit the passage in and out. These are nuclear envelope is a special kind of plasma membrane. The only difference is this one is just for the nucleus, and plasma membrane is for the whole cell. This is the structure of the nucleus, and as you can see at the center is the nucleolus. Just take a moment to look at the structure of the nucleus. Ribosomes, I want you to remember this, they carry out protein synthesis or translation, messenger RNA translation. This happens in the cytoplasm. We found them both in eukaryotes and prokaryotes. They are composed of two subunits. These subunits, we call them the larger and the smaller subunits. They are a mix of proteins and ribosomal RNA, and they receive messenger RNA as an instruction sequence of amino acids in a polypeptide. We said that already. Messenger RNA comes from the nucleus, and these ribosomes convert the messenger RNA to a polypeptide or a protein. In eukaryotes, some ribosomes free in cytoplasm. Yes, ribosomes could be free floating, or they could be attached to the endoplasmic reticulum. Some are free floating, others are attached to the endoplasmic reticulum, especially the rough endoplasmic reticulum. This is how the nucleus looks like with the ribosomes and the endoplasmic reticulum, which is this one. The endomembrane system. The endomembrane system has four components. One is the nuclear envelope, or the outer portion of the nucleus. And then we have the endoplasmic reticulum, which includes both the smooth and rough endoplasmic reticulum. And the third one is Golgi apparatus, and the last one is vesicles. And it helps to compartmentalize the cell. That means to divide the cell into certain kinds of dividing a cell into cells. I don't know if that's the right way of saying it, but it compartmentalizes the cell like in a prison cell. It compartmentalizes them so that a certain kind of reaction takes place only in particular region, not somewhere else. Transport vesicles carry molecules from one part of the cell to another. Is the endomembrane system. Endoplasmic reticulum is a very complicated system of membranous channels and saccules. Saccules are just sacs within a cell. They're just a form of saccules that might carry protein, carbohydrate, anything that the cell needs. And it is continuous with the outer membrane of the nuclear envelope. And it has two forms, the rough and smooth. The rough is studded with ribosomes. That means ribosomes are stuck on the rough endoplasmic reticulum and modifies proteins. Protein modification takes place here and forms transport vesicles going to Golgi apparatus. And the smooth endoplasmic reticulum, as the name implies, it is smooth because it has no ribosomes studded on it. And it is smooth, in a part it is smooth because it actually synthesizes lipids like phospholipids and steroids. As you know, lipids are very smooth by nature, like fat, or think of butter, it's very smooth. That's why it helps you to remember smooth endoplasmic reticulum synthesizes lipids. I'm not saying it is smooth because of this. I'm just saying it just looks smooth because it has no ribosomes. And on top of that, lipids are smooth, so they're related. I just want you to remember it that way. Their function depends on the cell. Some of them produce testosterone, others detoxify drugs, or others still do both or even more tasks. This is the endoplasmic reticulum. Golgi apparatus. Golgi is a stack of flattened saccles. As I told you, saccles are just biological stacks. And this is the transfer station. It receives vesicles from the endoplasmic reticulum, and it modifies molecules within the vesicles, and then sorts and repackages for a new destination. Okay, Golgi apparatus is just your UPS. This is the place where repackaging and sorting of all the vesicles takes place. And you'll put a tag on them, and then you send them where they need to go. Sometimes you just tag them and send them to endoplasmic reticulum, or sometimes you send them to plasma membrane. And actually, some of the saccles we find in Golgi apparatus are lysosomes, and lysosomes are digestive enzymes. They digest molecules. They're like a mini stomach for the cell. They digest a lot of molecules. And a genetic disorder that's associated with lysosomes is Tay-Sachs disease. And this genetic disorder has a lot of abnormalities associated with it. That means if the cell is unable to digest some of the molecules it doesn't want, there will be an accumulation of toxic chemicals within the cell, which might have a detrimental effect on the whole function of the cell. This is the endomembrane system, starting from the nuclear envelope, then the rough endoplasmic reticulum with the ribosome studied on it, and then smooth endoplasmic reticulum, and then the Golgi with light blue color. And then we have the lysosomes that digest the molecules. Yeah. Vacuole, I want you to remember it as this is a water station. These vacuoles are membranous sacs. They are larger than vesicles. They get rid of excess water. And you know water is really important for digestion. So vacuoles are also important for digestion. And in other organisms, vacuoles are used for storing plant pigments and animal adipocytes. Adipocytes are fat droplets. So some animals use vacuoles to store their fats for later use, so that they can use it when they needed it most. These are the vacuoles. Vacuoles are very common in animals and plant cells. Energy-related organelles, in fact, the only energy-related organelle that we know of is the mitochondria. For both plants and animals, and for plants there's another energy-related organelle, which is the chloroplast. So mitochondria, found in both plants and animals, are usually only visible under electron microscope, and is bound by a double membrane, very complicated double membrane. And this is the place where ATP is produced, adenosine triphosphate is produced from the breakdown of carbohydrates, and cellular respiration takes place here. And as you know, cellular respiration is the opposite process of photosynthesis. In photosynthesis, you use carbon dioxide and water to produce glucose and oxygen. In this one, you use glucose and oxygen to produce carbon dioxide and water. The inner membrane has folds that are called cristae, and the cristae are really important to increase surface area within the mitochondria. The inner membrane closes the matrix, and the matrix is where most of cellular respiration takes place. ATP synthesis takes place here. And one unique character of the mitochondria, which might appear on the MCAT. In fact, most of the MCAT practice exams have a question like this one. And the mitochondria has its own DNA and its own ribosomes, and mostly it is independently functioning without any order or instruction from the nucleus. Chloroplasts use solar energy to synthesize carbohydrates through the process of photosynthesis. Plants and algae contain chloroplasts, unlike the mitochondria. They do have 3 membranes. Mitochondria has 2 membranes. So the double membrane encloses stroma, and then the thylakoids form the third membrane. The thylakoid membrane contains pigments that capture solar energy, and like the mitochondria, chloroplasts contain their own DNA and ribosomes, and usually they are independently functioning. This class is a human biology class, and I might not ask you to know the plant structure in detail, but it is always a good idea to have a clear view of what chloroplasts are and how they relate to mitochondria, or how they appear similar with the mitochondria. This is the chloroplast structure. This is the double membrane system at the bottom and on top. And within this is the stroma. That's the clear part within the double membrane system. All the green stalks you see are the thylakoid membrane. And the green stuff you see is the granum. Thylakoid space. The thylakoid membrane has its own membrane. Okay. Yeah, for a moment I forgot I was recording the video because I was going into detail. Okay, this one is the mitochondrial structure. I'm gonna move this one down. The cytoskeleton and motor proteins. The cytoskeleton, you can think of it as the subway system of the cell, and at the same time, all the buildings of the cell. Because the cytoskeleton is all the network of interconnected protein filaments and tubules that you can use for transport and for maintaining the shape of the cell. And it extends from the nucleus to the plasma membrane, like the subway extends from the center in Manhattan to the rest of the boroughs. And we usually find the cytoskeleton in eukaryotes because prokaryotes are simple organisms and they don't need all this cytoskeletal structure, but eukaryotes are complex organisms and they do a lot of transportation in and out of the cell, which necessitates the presence or the existence of a cytoskeleton. Maintains cell shape, and there are motor proteins. These are the trains and buses of the cell. They allow the cell and organelles to move. We have the myosin, the kinesin, and the dynein. They are instrumental in allowing cellular movement. Myosin is really important in muscle contraction. I'm sure you will come across this biological protein when you talk about cardiac failure or heart disease, because it interacts with actin and it is like at the center of all kinds of muscular contraction. And it allows cells to move in a mobile fashion, the way an amoeba moves. Kinesin and dynein, how I remember this is kinesin it's like kinetics. You have to move. When you move, you have to move away, usually. And dynein is like dial in, and you are trying to come in. That means kinesin, it usually moves away from the nucleus to the plasma membrane, whereas dynein, it comes in to the cell. from the plasma membrane to the nucleus. But they both move along the microtubules. Microtubules are the train tracks of the cell. They transport vesicles from the Golgi apparatus to the final destination. That's what they do. Okay, so actin filament moves, not the myosin. Myosin has a head, see? It just stretches and, for example, it might contract this way and then it might stretch this way. In doing so, it is actually pushing the actin filament to go this way or that way. And this is the myosin filament. This is the microtubule or the train rack. It says microtubule, and this is the vesicle, this is the kinesin, and it is moving along the microtubule. Vesicle moves, not the microtubule. Microtubules are not actually like this. They look cylindrical, but they are not like this. Their actual structure doesn't look like this. They are cylindrical. They look like small balls coming together. They are small hollow cylinders. Their assembly is controlled by centrosome. They help maintain cell shape, and they act as a track, like a train track for organelles and other materials to move. And we have intermediate filaments that are intermediate in size, and they are rope-like assembly, and they run from nuclear envelope to plasma membrane. Intermediate filaments, just remember, these filaments are filaments that make up the collagen and elastin that are found in the extracellular matrix. So intermediate filaments, elastin, and collagen. Microtubules, they are small hollow cylinders. An example is cilia and flagella are microtubules. This is how microtubule actually looks like. It doesn't look like this. It is not like this flat. It actually looks like this, with the orange, the yellow and the orange alternating, because it has an alpha unit and beta subunit that are alternating, but it is hollow inside. It's like any kind of tube. Actin filaments, these are two chains of monomers. Actin filaments are like the DNA helix that are twisted in a helix. They form a dense web to support the cell. So we saw 4 kinds of filaments. We saw microtubules, we saw intermediate filaments, and we saw actin filaments. Microtubules are the microtubules are the cilia and flagella. Intermediate filaments are the elastin and collagen, and actin filaments are over here. Centrioles are made of 9 sets of microtubule triplets. 2 centrioles, they are triangles in animal cells. We find them in animal cells, but not in plant cells. These words, the centrioles, we are going to see it in our next Zoom recording, because centrioles are really important in green cell division, because this is where the centrosomes come into play, whereby the microtubules are extending to the opposite poles of the cell to detach the chromosomes during anaphase. These are the centrioles. Cilia and flagella, we find them in eukaryotes, and they're really important for the movement of the cell and fluids past the cell. They have a similar construction in both, which means 9+2 pattern of microtubules. Cilia are shorter and more numerous than flagella. The 9+2 comes from here. We have 2 central microtubules, and we have 9 microtubule doublets. That's what the 9+2 is about. Cell walls, plant cell walls, we have primary cell walls and secondary cell walls. The primary cell walls are the cell walls we are familiar about. It's made of cellulose, which is a polysaccharide made of glucose. The wall stretches when the cell is growing. The secondary cell wall is not common. Only woody plants contain a secondary cell wall, and it forms the inside primary cell wall. And then we have the plasmodesmata. Plant cells connected by numerous channels that pass through the cell walls. Okay, plasmodesmata is, think of it as, let's say you have a series of cells, like 5, 6 cells. And then if you connect all the cells by a tiny wire, this tiny wire is serving as a plasmodesmata because it enables you to move certain chemicals, water, and other important nutrients to move from one cell to another. This is the extracellular matrix. This is the matrix that you find outside of the cell. It's made of elastin, collagen, various polysaccharides, receptor proteins, various proteins. The exterior cell surface in animals, animals don't have cell wall. Rather, they have extracellular matrix, which is a meshwork of proteins and polysaccharides. We said that already. Meshwork of polysaccharides, and then we have proteins, collagen and elastin. Collagen and elastin, they are well-known proteins, and the matrix varies from cell to cell. For example, if you take a bone matrix, it is very strong because it is a bone. But other cells, the matrix is not that strong. Cell wall provides support to cells in many animal cells, plant, fungi, and protists. We find cell wall in these organisms. The extracellular matrix is what we said that already. Collagen, it resists stretching because collagen is very strong. Collagen actually makes up the bone. Elastin, it is elastic as the name implies. When something is elastic, it gives you resilience. It is not rigid, which is good. Junctions between cells of the intestinal wall. I don't think you need to know all of this. Adhesion junctions, these are junctions between cells. This is internal cytoplasmic plaques joined by intracellular filaments. Just know that adhesion junctions are junctions that are found between cells because cells cannot function independently. So it is a sturdy but flexible sheet of cells. It's not just two cells, it has a lot of cells coming together and they're interacting to each other. Tight junctions are, as the name implies, they're tight. They are impermeable barrier because you don't want just every chemical to just move from one cell to the other freely, because at the end of this day, a cell is like a prison cell. Only a certain individual or certain chemical reactions take place in that cell, and you don't want chemicals to move from one cell to the other and contaminate the reaction. Gap junctions also allow communication between cells. What's happening in gap junctions is you are trying to connect the plasma membranes of the cytoplasm of two cells, but to do so, you try to connect the plasma membranes through a channel. So gap junctions are channels that connect two plasma membranes together. Okay, that's all. What I want you to remember is I'm not gonna ask you like a detailed question, but definitely know the big picture questions. Even though it is a human biology class, I still might ask questions about the cell structure of plants, just at least in terms of to enable you to see the comparison between animal cell and plant cell. So just focus on the big pictures, and yeah, you should be fine if you do that. Right? Thank you.